Welcome, and thank you for attending our webinar on managing human factors and reprocessing of reusable devices, validation considerations. Here to present is Emily Mitzel, our resident expert here at Nelson Laboratories. Emily Mitzel is a laboratory manager at Nelson Labs, a microbiological testing company specializing in improving the quality of life by ensuring medical products are safe, sterile, and functional. Emily's expertise includes reusable device reprocessing validations, pharmaceutical testing, microbiology testing, and media production. Emily presented at the AAMI FDA Reprocessing Summit in 2011, facilitated at the AAMI FDA Roundtable discussion in 2013, and currently presents at Nelson Laboratories and other seminars. Emily is an active committee member of many working groups with the Association for the Advancement of Medical Instrumentation and American Society of Testing and Materials. Emily holds Bachelor's of Science and Master's of Science degrees in microbiology. Welcome, Emily. The time is now yours.
Hello, and thank you for joining us for this webinar on managing human factors reprocessing of reusable devices and all of the validation considerations associated with this information. Reusable devices come in many types, sizes, shapes, and complexities. The problem with reusable devices is that the cleaning and disinfection and sterilization of these reusable devices in the healthcare facilities. There have been many issues with in ensuring that these devices are cleaned, sterilized, and disinfected appropriately. There have been many issues with finding devices that have not been cleaned sufficiently, and these have been shown in some of the information on the news and websites and human-acquired infections. Here we're seeing some human tissue found in an arthroscopic cannula during an infection outbreak investigation. This next picture shows a bristle, and so the device was appropriately cleaned, but there was a bristle that came off of the brush that cleaned this device, and it was left inside of the device. These devices show devices that are packed with debris that were utilized in procedures. This link is a presentation that was given by the Today Show that investigated dirty surgical instruments. There have been a lot of news lately regarding healthcare associated infections, and they estimate that these cost us about $9.8 billion annually, and approximately 100,000 people in the U.S. are killed by healthcare acquired infections. So this shows the great need to make sure that we are performing these validations and making sure that we are cleaning, disinfecting, and sterilizing devices appropriately in the healthcare facility. In today's talk, we will be going over guidance for the reusable device manufacturer, some information on a roundtable discussion regarding how clean is clean that occurred in September of 2013, some information regarding device design, human factors associated with the cleaning validation and also the sterilization validation, instructions for use information, and some manufacturer's responsibilities and considerations. There is some important guidance that has come out in the past couple of years. The draft guidance for industry and FDA staff came out as a draft in May 2011. Steve Turtell from the FDA just discussed last month, or a couple weeks ago, that they are still working on this draft guidance and have gone through all the comments from industry and staff and are hoping to make this in a non-draft format very soon. AAMI Tier 12 was updated and revised in 2010, and AAMI Tier 30 was updated and revised in 2011. This is very pertinent information in validating the cleaning, disinfection, and sterilization of reusable medical devices. There are a lot of other really important things going on with the AAMI working groups related to reprocessing. There are a lot of working groups that are associated with the validation of these endoscope reprocessing, human factors, quality systems, which takes into account what is performed in the healthcare facility, and so on. A few new working groups from AAMI are the human factors device reprocessing. That is in a draft version, and it is going to vote very soon. Standardized instructions for use is a draft, but it's kind of on hold due to getting some information from other countries and so on. The quality systems for device reprocessing is in an outline at this point and is working on getting this into a draft format. The endoscope reprocessing document had over 200 comments that we went through a couple weeks ago, and we are getting all of those incorporated into the document. Managing sterilization of loaner instrumentation is in a draft format, and low intermediate level disinfectants and their use, that's more of an outline format at this time. There's also a couple working groups from ASTM that are working on validation requirements. The one that we are going to be working on next week in Toronto is the new guide for standard test oils for validation of cleaning methods for reusable medical devices. And in this document, we are going to be adding in test soil information and recipes on what is recommended to validate medical devices. In September 2013, we had a roundtable discussion on how clean is clean. The participants of this discussion were AAMI staff, FDA staff, medical device manufacturers, some test labs, and some hospital staff. What we were trying to do is really determine the maximum amount of residual soil that is appropriate, that can be left on a medical device and still render it safe for patient use. This issue was identified as one of the 7 clarion themes to come out of the AAMI FDA summits that occurred in 2011 and 2012. So really, cleanability is determined by device design, human factors, instructions for use, and compatibility of the device to the cleaning and sterilization instructions. We ensure clean devices by validating, which means manufacturers need to validate the cleaning method that will render the device safe for patient use, and then healthcare facilities need to verify that that validated procedure is effective and can be performed in a healthcare facility. We have determined that there is not one test oil that can be used on all medical devices. We need to determine what test oil is used by how the device is utilized in a clinical setting. Another idea that we have determined is there needs to be an increased research performed on how to determine how healthcare facilities should verify cleanliness after the cleaning procedure. This includes the testing methodology and frequency. Right now, there are many medical device manufacturers that have performed hundreds of cleaning validations on all of their medical devices. And if we could put this information together and really determine what we consider to be a cleaning efficacy number and so on, that would really help in the future of cleaning validations for reusable medical devices. We also are working on putting together a position paper that would really kind of highlight this information by either performing more research or roundtable testing for medical device manufacturers. Also, what we need to do for this implementation is we need to change technical information reports like TIR-12, TIR-30, and also changing different standards like ST58, ST79, etc., to incorporate this information to make sure that we are all on the same page on how clean is clean. One thing that we need to think about are in the instructions for use. Now, these need to be very standardized, and it would be very good for them to be grammatically correct, very legible, presented in a logical order. So we need to make sure that we are performing them in a step-by-step process. All the instructions need to be short and concise. All the information needs to be spoken in a passive positive and active voice. We need to number the steps in a numerical format. And it's very good to show images, show essential steps in images so that it's easier for people to understand. Some things that we do not want to have in IFUs are words like a minimum of, if appropriate, if possible, and if necessary. This language has been discouraged and is no longer acceptable. We want to make sure that we are very specific in what we are stating on the IFUs. And so we want to state very specific ranges and explain how things are done in a specific manner. Disassembly and assembly instructions have become very important in the IFU. This is necessary in some devices to make sure that devices are cleaned properly. This may include illustrations, diagrams, and also videos to show healthcare facility workers on how to perform these steps. The specific instructions in the IFU needs to include the specific detergent dose, temperature of each step, the water quality, the time to perform each step, the brush type and size, and any specialized supplies, which includes the part numbers of each of these supplies that are needed in the cleaning procedure. Human factors plays a huge role in the sterile processing because this is performed by people, and we want to make sure that all the information regarding training, competency, and documentation are performed very specifically for the healthcare facilities to make sure everybody is trained on how to perform the cleaning validations of these medical devices. The quality system standard, AAMI ST90, is just started, and the first draft is just being completed and it wasn't complete by the spring 2014 meeting, but it is being worked on at this point. It is specific for guidelines for procedures, documentation records, and so on for reprocessing reusable medical devices in healthcare facilities. It includes audits, process improvements, performance testing, education, and competency. Some new expectations of the FDA for medical device manufacturers is to provide on-site training and education for sterile processing personnel. This opens the communication and encourages collaboration on medical device designs that meet functional requirements and also addresses human factor considerations. This is very important to really open the dialogue to healthcare facilities and medical device manufacturers to ensure that the instructions that they have put forward for the healthcare facilities are appropriate and are easy to follow. As part of this, medical device manufacturers need to consider feedback from healthcare facility personnel, sales reps, etc. They also need to solicit input from individuals knowledgeable in the processing equipment and methods to ensure complete and accurate analysis. Medical device manufacturers may help develop education, training, and competency verification materials for healthcare facilities. And we also need to keep in mind that proper PPE for healthcare facilities needs to be used during cleaning when performing the validation. In conjunction with designing devices, we really need to think of items with the medical device that may have issues in performing functional yet cleanable and sterilizable product designs. We find many engineers really thinking about the use of the product and making sure it is fully functional for what the surgeons need and so on, rather than really making a device that is specific for cleaning, disinfecting, and sterilizing. So we really need to be thinking about this part up front along with designing the device for functionality. Some design features that make cleaning difficult are braided and twisted wires, textured surfaces, hinges, springs, dead end lumens and channels, inaccessibly cracks or crevices, any type of mated surface, sharp internal corners and angles, or any type of O-rings. All of these parts may harbor unwanted organisms and/or organic material and may make these devices very difficult to clean and sterilize. Another material design feature that may make cleaning difficult is the type of thermal conductance on the device. Aluminum-based materials could be an issue, as well as pliable materials such as silicone or rubber. Here is a temperature profile example of a medical device. You can see here that the product is a dark blue line, and it is not quite at the highest temperature of the chamber during the first pulse, but as you can see in the second pulse, it is almost to the temperature that the chamber is, and by the third pulse and the exposure time, it is right on the appropriate temperature. This differs for the silicone device. As you can see here, again, the blue line for the product does not meet the temperature required until after the exposure part has started. So it's ramping up along with the chamber temperature, but the chamber temperature is a lot higher, and the temperature of the silicone device does not even get up to the appropriate chamber max until about halfway through the exposure period. Another part that may make components cleaning difficult is that we need to make sure that devices are reassembled properly. And if there are a lot of parts, they could be misplaced or intermixed with other devices. This can cause a lot of confusion, which may extend the processing time. And that is very difficult in healthcare facilities as they are processing hundreds of devices every hour. Complex devices give us a lot of unavoidable problematic design features. But as we know, we have developed very complex devices because of our need for surgical procedures. But we just have to keep in mind that these devices may require more stringent cleaning procedures, such as mechanical or automated cleaning methods. So as part of the cleaning validation, some considerations that we need to make are that we are performing worst-case testing, appropriate test soil, appropriate residual testing. There are several methods for contamination for the medical devices. We need to keep in mind that the contamination method that we utilize must be worst case for that specific device. So we need to think about the clinical procedure that's performed with the medical device, but we also want to make sure that we are not contaminating these devices to an extent to make it extremely difficult or near impossible to clean. We want to make sure that we are performing a worst case situation, but not a ridiculous case situation for the cleaning validation. So when we perform this testing, we determine what is the worst case dwell time and what is the worst case surgery time as determined by the device type. As you can see in this picture, there may be many medical trays awaiting decontamination. So we have to keep in mind that there is some amount of time that will take place between the surgical procedure and when the devices can be cleaned. During this time, we need to make sure that the devices are kept in a moist fashion if at all possible. There are many different types of methods of cleaning depending on the device type that is utilized. We need to make sure that we are performing the cleaning exactly per the specifications of the manufacturer's IFU. A manual form of cleaning is the most common method, and it is mandatory to validate a manual cleaning process. This may include soaking, brushing, and flushing the device. A mechanical means of cleaning may also be incorporated in the manual procedure or also incorporated into a pre-cleaning prior to the automated cleaning. An automated cleaning procedure is also mandatory if the device can handle an automated cleaning procedure. In this case, a washer disinfector is utilized and certain detergents are placed into the detergent, or excuse me, into the washer that's appropriate for the washer type and the medical device. Some challenges regarding cleaning instructions are when sterile processing personnel must alter their process because of unique instructions in the IFU. There are many medical devices that have such complex instructions that it is very difficult for healthcare facility workers to go through every step of the process. Manufacturers are encouraged to use basic cleaning parameters and also pre-programmed washer cycles. In determining what detergent should be used with a validation, we need to make sure that the detergent is compatible with the entire device. We also need to ensure that this detergent that is validated is available to the end user. So that really depends on where the device is marketed, whether it's marketed in the United States or Europe or some other country. We also need to consider the clinical conditions in which the device will be cleaned in the healthcare facility, and also use the recommended concentration on the detergent itself. So what the FDA has specified is that if a detergent specifies to use 1/4 to 1/2 ounces per gallon, we need to make sure that during the validation we stay in this range, the IFU stays in this range, and also for the validation we want to use the lower quantity of detergent to put it more of a worst case situation for the cleaning validation. Along with the type of cleaning procedures, we need to use all the supplies that are appropriate to each cleaning device. We need to ensure that they are compatible with the device design, that they aren't going to hurt the device at all during the cleaning procedure. If there is a specific brush that is specified to clean the device properly, it should be supplied with the device, or a part number should be given so that the healthcare facility can order it appropriately. After the cleaning procedure is performed, a visual inspection of each device needs to be performed. Now, depending on what device type is being inspected, there are different methods of the visual inspection. First of all, an inspection by the naked eye is the most important, to check every crack and crevice that can be visualized by the naked eye. Next, we need to determine if a borescope should be utilized to test inside any lumen or internal aspects of any of the device to ensure that there is not any visible soil inside or outside of the device at all. Cleaning markers for the cleaning validation are based on what the device comes into contact with and also what the FDA is accepting as the most appropriate soils for the validation. At this point, the most popular and appropriate markers for testing the validation are protein, hemoglobin, and total organic carbon. Some other additional markers that can be utilized are a bioload reduction, which is appropriate if selling the devices in Europe to get the CE mark. Also, testing for carbohydrates may be utilized, as well as testing for any detergent residuals, as well as cytotoxic properties of devices, and also endotoxin testing. The acceptance criteria for cleaning validation is kind of difficult at this point. There is really no established performance criteria for these tests. The TIR 30 has some benchmark criteria for endoscopes, but all of these specific criteria need to be determined and justified by the manufacturer. These need to be stated prior to the validation and must be met during the validation. The cleaning information needed to perform the testing is we need a description of all accessories required for the cleaning, the specific techniques used, including rinsing, the type of water quality that is used for each process, the concentration and type of each chemical and detergent that's utilized in each process, and the exposure time and temperature of each step. So now moving on to a sterilization validation for reusable medical devices, we're going to be mainly focused on steam sterilization, as that is the most utilized method of sterilization in a healthcare facility, but healthcare facilities also could use sterilization using STERAD, ethylene oxide, dry heat, and liquid chemical, which is probably the least utilized at this point. So to perform sterilization validation, we need to perform a sterility assurance level validation with half-cycle parameters, then a dry time validation with full-cycle parameters, and then temperature profiling for case studies. Biological indicators will be placed in the most difficult to sterilize areas and also a lot of representative areas within the tray. Some of the most difficult to sterilize areas include lumens and mated surfaces. For these biological indicators, it is preferred to use a biological indicator rather than so inoculated carrier rather than direct inoculation. The acceptance criteria for a sterilization validation is that all media used for the sterility testing needs to demonstrate growth promotion. All validated cycles must be within specifications. The cycle tape must show all of the parameters that are appropriate for the cycle. We must kill off a 6-log reduction in the half cycle. Packaging and products must pass visual inspection with no moisture observed, and the packaging weight gain cannot exceed 3% to maintain sterility. As part of the sterilization validation, we need to use materials that are all FDA cleared. These include the sterilizers, all the accessories, which include biological indicators, chemical indicators, and all the packaging associated with the sterilization validation. The parameters validated for these medical devices must come from AAMI TR12 and AAMI ST79. The FDA is not accepting extended cycles for sterilization validations, as the healthcare facilities must be able to perform the sterilization parameters that are validated within the process. Here's an example of the sterilization parameters in AAMI ST79. The most commonly accepted cycle parameters is 132 degrees Celsius with a 4-minute exposure time and a 20 to 30-minute drying time. We have found that the extended drying time is acceptable in many cases. Even our local healthcare facility utilizes a 55-minute dry time because that is what it takes a lot of times to make sure dry time is adequate for all of the large trays associated with the surgical procedures that are performed. Some sterilization considerations include the device design. We need to make sure that the material of the devices heats up quickly and remains hot throughout the exposure period. We need to make sure that we limit any blind holes or dead-ended lumens, and that cannulas and entire devices allow for air removal and steam penetration. It is also important to make sure that the trays do not contain more than 25 pounds of mass. This only does not make it difficult to sterilize, but it makes it very difficult for healthcare facility workers to move the tray in and out of the sterilizer, as well as moving it around the healthcare facility. Some other considerations include tray layout, which makes it very important for holders to have limited contact with the device and to make sure that steam is not blocked with these holders. We need to make sure that devices are in an unlocked and open position, and we also need to make sure that the mass is distributed throughout the tray. We also need to ensure that the devices are not stacked on each other and that they are in a fashion so that as the sterilization process occurs, as in steam sterilization, it is in a fashion where if there's any water that's dripping from the devices, it drips down the device and off of it through the drying procedure. During the dry time testing, some things that to keep in mind are that materials that stay hot do the best in ensuring that the dry time testing is appropriate. Reducing mass may help with the dry time testing also, and we need to make sure that all surfaces do not allow any water to pool and that everything allows it to properly drain. The packaging material is also very important to ensure that the products can appropriately dry during the drying procedure of the sterilization cycle. Some sterilization validation information needed according to ISO 17664 is we need to describe the exact techniques to be used during the sterilization process. We need to describe any accessories that are utilized, and this includes the packaging. In describing the process, we need to have the exact concentration of the sterilant, the temperature of the sterilizing agent, the humidity for the sterilization process, the minimum exposure time, the pressure, and the post-sterilization activities. So one example of this is how the trays are removed from the sterilizer. Some things to think about in thinking about human factors in the healthcare facility is that we should expect some amount of error use anticipated in the design and of the device and its labeling. We need to ensure that there is correct competency assessment for all personnel performing cleaning and sterilization of reusable medical devices. And this is ensured by making sure that the education, experience, and training is well documented. Medical device manufacturers should work with healthcare facilities to ensure that this is all occurring and also to make sure that all the cleaning procedures are easily followed. Thank you so much for attending this webinar conference on managing human factors in reprocessing of reusable medical device. This is all the validation considerations that should be taken into effect when performing a validation for cleaning reusable medical devices. Please let us know if you have any questions. You can reach us at nelsonlabs.com, and you can contact us with any questions or clarification or any other information that you may need. Thank you very much. Goodbye.